All right, welcome everybody. We're excited to continue our FAFP webinar series this afternoon in lieu of our annual educational conference this year. We certainly appreciate the great turnout and really express our gratitude to our members and sponsors for their continued support. Just to start off with a few housekeeping items, please keep your video and audio off throughout the duration of today's program. We'll ask David to answer questions from the audience at the end of his presentation, and we have a team of volunteers who will be monitoring the chat box and facilitating your questions with David. So please do enter any questions you have directly into the chat box. We'll also be recording the program to post on fafp.net, which has recently been revamped, so please check that out. Our speaker today is Dr. David Buckley. Dr. David Buckley is the Director of Technical Consulting for the retail and food service sectors at Diversity Incorporated. David is responsible for consulting with consumers on food safety, sanitation, and infection prevention and control programs. Additionally, he liaises internationally with marketing and R&D to guide research and product development for value-added programs, as well as leads on and conducts independent research. Before joining Diversity in January 2020, David was a postdoctoral research microbiologist at the United States Department of Agricultural Research Service. His focus was investigating a novel bio-based chlorine dioxide gas packaging system and other intervention technologies to improve produce safety against Listeria monocytogenes, Salmonella, Escherichia coli, O157H7, and hepatitis A virus. David earned a Bachelor of Science in Biology and Chemistry from East Carolina University in 2011 and a doctorate in microbiology from Clemson University in 2017. His background is in applied microbiology and food safety. He is actively involved with IFP, GFSI, CFP, and other industry associations. And so with that, David, I'll hand it over to you.
We'll jump right into it. I've already done opening remarks, but first I'd like to go over key terminologies, do a review on immunity, then we'll look at SARS coronavirus 2 by the numbers, discuss what we've learned so far, talk about the vaccines, and then talk about these variants that are obviously in the news a lot and are causing some angst. So with that, I'd also like to pause and do a shout out. If you guys don't listen to This Week in Virology, it is an excellent podcast. I listened to it before COVID, but during COVID, it's been awesome. It's almost like my own little personal journal club. So if you haven't, I would take a listen to it. They talk about a lot of great things, and it's kept me sane throughout the pandemic. So with that, we'll go over some key terminologies and immunology review. All right, so these are some terms that you may not be familiar with or may have heard before, but aren't exactly sure what they are. And they pertain directly to this presentation and to coronavirus in general. So antigen and antibody, these are two terms that are used quite frequently when discussing vaccines and immunity. And antigen is basically any substance that can induce an immune response. So think of an allergen. So when that allergen gets into your body and if you're allergic to it, you generate an immune response to that specific allergen. An antibody is an immune-related protein that binds to that specific antigen. So when that antigen stimulates your immune response, there's a number of things that happen, but one of them can be that an immune-related protein called an antibody interacts with that specific antigen. The other one is efficacy and effectiveness. These two terms are often interchanged when they shouldn't be, kind of like the terms disinfectant and sanitizer. Those are two mutually exclusive words that shouldn't be interchanged, but are often used interchangeably. But efficacy is really us understanding what is the performance under ideal or controlled conditions. So if I'm testing a sanitizer in a laboratory versus understanding how it works in real world conditions. And so that's where effectiveness comes into play, because you can see differences in performance. So performance under ideal conditions and effectiveness is performance under real world conditions. Herd immunity, I just put this up here, but we're going to talk about that in the next few slides. So I'll skip over it. mRNA, this popped up a lot because of the Pfizer Moderna vaccines. Messenger RNA, or that's what the M comes for, is message, is a nucleic acid courier, and it basically encodes the genetic message for creating a specific protein. And your molecular machinery within your cells translate this into a protein, and is the basis for those Pfizer and Moderna vaccines. Reactogenicity, this is actually a term that I recently learned, but is important because people are really worried about the vaccines and what kind of symptoms you might get from it. But reactogenicity is something that we refer to as common and expected reactions after vaccination. So if you think of your flu vaccine, you might feel crummy for a few hours after it, or maybe a day. That's not something that you should call up the CDC for and say that you have an adverse event. If you just kind of feel crummy for a day or two, that's common. And examples of that are maybe a slight fever or sore arm. And so we refer to that as reactogenicity and is typically common. These three down here, virus isolate, variant, and strain, you'll notice that the news and even scientists will often interchange these three, but they are three distinct terms. So a virus isolate, this is usually collected from a patient, but can be from other places like an environment. It's just a specific virus that's been isolated from an individual, and they've characterized it. That's kind of the whole purpose of this, that's been isolated. And that's what a virus isolate is. A virus variant, so this is something that is genetically distinct from what we would refer to as the wild type or a predominant virus that's currently circulating. And that's what we're seeing right now is variants of concern, and they're not technically strains yet. We're not really sure what to do with them, so we're classifying them as variants of concern. A strain is just a much higher bar. If you want to be considered a virus strain, then that isolate or variant that you've identified needs to demonstrate a new biological property different from the progenitor, and that could be, in this example, higher stability or increased transmissibility, increased virulence. What we've seen so far among variants is that there's suspected, but nothing completely confirmed. And so that's why we're still referring to them as variants. All right. And so if we talk about, oh, here we go. Yeah, so next is immunity. So when you talk about the vaccines, there's really two different ways to develop immunity. There's immunity to disease, so meaning I get vaccinated and I no longer will present disease. And then there's immunity against infection. So here, what we think is happening with SARS coronavirus 2 and what we can demonstrate so far is efficacy against disease. Those were the primary endpoints of the vaccines, and not the, and so infection hasn't really been elucidated yet. So we're not really sure how well these vaccines that we're using under EUA, or emergency use authorization, we're not sure how well they're doing at preventing infection. And that's something that vaccine companies and the CDC are very curious in, and they're currently looking at, but we don't know that answer yet. All right, and so for herd immunity, this term gets thrown out a lot, and you hear people keep saying, oh, we need 70% or 80%, and maybe it varies by country. That's true that it varies by country, but herd immunity is a bit more nuanced in its definition. So if you think about what people typically think of herd immunity, that means you look at these pictures here at the bottom, no one's immune, people get sick, and you get this big outbreak. But if you go over to the far right, you see that all of these yellow people are immune, or they've been immunized, and so that prevents the spread of the disease. Now, this is kind of a model for sterilizing immunity, meaning that you are protected from disease and infection. But most diseases, or viruses, I should say, because we're talking about viruses, don't really provide that. But the two that I can think of are maybe hepatitis A and measles. Measles, for instance, you get lifelong immunity from infection and disease. There's about a 97% chance of that happening. So, but for coronavirus, we don't think that we're going to get to sterilizing immunity, but we will get to hopefully a point where people have been exposed to the virus through a vaccine. So they're getting immunized via vaccine route, and they are, I guess that they will be protected from the disease portion, which will drive down hospitalizations. That's kind of the key endpoint that we're really attempting to deliver at this point. So when people talk about herd immunity, there's a lot of nuances associated with it, and people will probably argue about what I'm saying about herd immunity, but there is nuanced definitions, and that's just something to keep in mind when you throw that term out. All right, and so I guess I should say that we do have a formula for calculating what that should be, and you can use the reproductive effective number. So if you say SARS coronavirus 2 has a reproductive effective number of 3, then the herd immunity threshold is 67%. But keep in mind, this number is not static. It is dynamic and can change based on the population. All right, and so so that's what we need is for population immunity, but what do we need for good individual immunity? Well, there's two parts of our immune system. There's the innate immune response that happens very quickly after introduction. And then we have the adaptive immune response that is a bit slower and takes a little bit longer to come on board and start working. But what we really need is for our adaptive immune response to jump in and start making a difference. And that's where we want to see our B cells start to differentiate and start creating antibodies, or the plasma B cells. And then we also want some memory B cells so that we can respond in the future. And then we also need the T cells to help out. They get talked about less, but they're also very important because they help train our B cells in creating more specific B cells and better response. All right, and so for SARS coronavirus 2, by the numbers, this is kind of where we're at. Back in July, I did a similar presentation in July, and this is where we were at. It is astounding to see where we were at in December and where we're at today. It's pretty crazy to see. If you asked me back in January if we would be at this number, and I would have thought you were crazy. But I would also say you're crazy if you told me we were going to have 65 million people in the United States have a vaccine put into their arm against this virus. But this is where we're at today for vaccines administered in the United States. It's really quite remarkable what we as a society have come to. But the point here that I'm trying to drive home is that right now we would love to get rid of infections and symptomatic illness, but the key thing to drive down right now is hospitalizations. We really need to get these numbers down, and then hopefully, fingers crossed that it will protect against infections. And also, as food safety professionals, we know that what is reported by the CDC is often underestimated. We know that people don't always seek help and don't always get tested. And so we know that the numbers that are reflected here for US cases is likely very, very small compared to what is truly happening out there. And this is just some numbers from February and September. The CDC estimates that there's 53 million infections of SARS coronavirus 2 that occurred within these months. So that's drastically smaller than this 28 million up here. All right, so what we've learned, there's really a lot that we've learned in the past year associated with this virus. At first we thought, okay, the quarantine guidelines are 2 to 14 days and we have to stick with that. But we've also learned that there's post-quarantine transmission risk. And so the CDC's updated their guidelines to state, well, okay, you can get out of quarantine under certain circumstances at day 10 or at day 7. And this isn't just mind games the CDC's playing, it's really based on science and data. And so you can end quarantine at day 10 with no tests and no symptoms reported, but you still carry a residual risk of, I think it's 1 to 10%. And same with day 7, if you do end your quarantine at day 7, you need to get a rapid test. But it can be ended at day 7 with a test, but you still have some residual transmission risk. And then for those people who have been vaccinated, so you received your second dose and it's 2 weeks after, you don't technically need to quarantine there. You have this window about 3 months, and just as long as you're asymptomatic since that current COVID-19 exposure. I suspect that this is going to change within the next 3 months. I think we're going to know so much more about immunity and how well the vaccines are protecting against infection. And whether or not someone who has been vaccinated is carrying the same viral load. Those are answers that everyone is seeking, but we don't have them yet. But if I was going to bet in Vegas, I would say that these will change in the next 3 months. And so mask data, I was also really skeptical in the beginning about masks, just regular cloth masks or the surgical masks, and whether or not they would do a good job at preventing respiratory droplet transmission. But there's been a lot of epidemiological information, and this is some papers that the CDC has cited that show the response of masks and really what we currently know about them. And I think it does show that masks do provide some benefit. They're not a panacea, but they do provide some benefit. So I don't want to spend too much time on this because hopefully everyone on this call understands that masks are important. But here's some of the science behind it that you guys can reference in the future. So transmission, this is obviously a hot topic. I put red as kind of the driver, yellow is a player, and green is low risk. So what we believe is the driver is contact and droplet transmission. Airborne and fomite transmission are definitely ways that the virus can be transmitted, although we don't believe that these are the true drivers. And then these these transmission routes are also ways that maybe it could happen, but they're clearly not a driver and low risk. And I'm definitely in the camp that foodborne transmission is very unlikely. And I just want to point this statement out too, that the FDA put, I think maybe a day or two ago, they are reiterating that the current epidemiological evidence and scientific information we have indicates that SARS coronavirus 2 is not being transmitted through food. Although I think the key point here is that it's low risk. I think it's unfair to ask a scientist, is it possible? Because anything's possible, but what's the real risk associated with it? And then so that's for transmission. But what can we do as individuals and as a collective to prevent the transmission of SARS coronavirus 2? This isn't a traditional foodborne pathogen, but it is affecting our food settings. And so what do we need to do to mitigate this? And these are kind of the key things that organizations can do and individuals can do to help prevent the spread of respiratory illnesses, specifically SARS coronavirus 2 and the flu. But those are maintaining health facilities, a healthy facility, having smaller facilities, preventing crowding, protective barriers, source control, cleaning hands. These are all very common things that we've come to know and understand. But knowing that you should do this doesn't mean that you can select one and pick it and that's all you can do. I put these hurdles up because everyone's familiar with the hurdle methods that we use in food safety, and that's kind of what we're doing here. You put more hurdles in the infection chain, and eventually you're going to break that infection chain. So applying multiple infection prevention strategies is the best approach. Just like I said, masks aren't a panacea. No one of these is a panacea. And we got to use a multi-pronged approach to control this in our environments. And so I'm going to spend the rest of the talk going through, viral replication period and the inflammatory period and what we've learned about SARS coronavirus 2 in this fashion. I really enjoy this figure. It's from a paper that was recently published on the 7th of February, maybe the 7th or 3rd, but it shows, what are the phases of this disease? It's such a complicated disease. It's really bimodal, and there's just a lot to dive in here. But I'm not going to go through this figure. You guys can take a look at it when you want on your own leisure. But so if we start at the viral phase, what do we know about this, and what can we do about it? There's a lot of antivirals out there that people are using. And then there's also some monoclonal antibody therapies that are out there. So I won't go through all of these, but these are some of the ones that are being used. I put hydroxychloroquine in red here because it is such a hot topic. I think that hydroxychloroquine showed some initial benefits, but overall, there's there's a lot of data that shows that it didn't actually perform well. And the next slide, I'll talk about one piece of that information. And then ivermectin is something that people are interested in, but the CDC doesn't, or sorry, I shouldn't say the CDC, but the health professionals are really not sure how ivermectin is going to perform as an antiviral. But talking about hydroxychloroquine, so there was an initial thought that hydroxychloroquine was going to work well, and that's because an idea of how hydroxychloroquine works is that you get hydroxychloroquine, the virus gets into your cell, and it raises the pH and it blocks cathepsin L, and so you don't get fusion. But what we've recently found is that SARS coronavirus 2 actually prefers another mediated, or it's TMPRSS2 mediated activation and fusion. And so this is something that hydroxychloroquine doesn't present. And so some of the initial studies used a cell line called Vero-E6 that expresses low levels of this enzyme TMPRSS2. And so because of that, SARS had to use this mechanism in the cell culture system, which artificially boosted the efficacy of hydroxychloroquine. But as we know, when you translate this to human trials, it doesn't translate well because human epithelial cells and in the lung, those cells do express high levels of TMPRSS, and so hydroxychloroquine was blocking an entry method that didn't really matter as much. So there's a lot of reasons, but this is one key reason. And the reason I present this today is that it's a relatively new paper that found this out. This wasn't something that we knew early on about SARS coronavirus 2. And so some other things we've learned, associations with disease severity and transmission. We've also learned that SARS coronavirus 2 can dampen the innate immune response. So this is something that SARS coronavirus 1 from 2003 didn't do very well. Originally, symptoms started to occur right at the onset of you being able to transmit the virus. And so we did a really good job at capturing those folks in the hospital systems and quarantining them and isolating them from everyone else. However, SARS coronavirus 2 has developed a new mechanism that dampens our innate immune response. I believe it's interferon's response. And so that allows it to gain entry, dampen your immune response, and then allow it to replicate before you start to show symptoms. And then by that time, you've already been infectious and been able to spread. And that's why we're seeing a marketable increase in the amount of transmission among those who are pre-symptomatic or asymptomatic. Another study, and I feel like this keeps popping up, is blood type. So blood type is clearly an obvious place to look. But I mean, there's disputed evidence that I think smallpox. Smallpox may have selected against blood group antigen A, and the plague may have selected against blood group O. And we do know that norovirus does have a preference for certain blood types. So those who have an inactivated FUT2 enzyme don't express that blood group antigen. They're resistant to some genotypes of norovirus. So clearly, obvious place to look. And the studies have been mixed here. This study from LATS in 2020 found that there was no specific blood type that was associated with elevated risk. There's been some newer studies that have suggested that certain blood types will prevent you from having severe disease. But I think the key thing here is that just because you have a certain blood type doesn't mean that you shouldn't get vaccinated and that you won't get infected. Relying on this alone is not a way to understand transmission or prevent disease. The other key thing that I thought was very interesting was vitamin D. So vitamin D in a few studies have demonstrated that if you have a vitamin D deficiency, you may be at an increased chance for severe disease. I don't think any of these were randomized controlled trials, but they do show strong evidence. And the fact of asking someone to take vitamin D is pretty low risk too. But I will say, don't go jump into vitamin D stock and throw all your money into that, because we're not exactly sure, but there's some good results out of vitamin D use. So reinfection, this is something that everyone is very interested in. What's my likelihood of being reinfected? There have been instances documented where people have been reinfected with SARS coronavirus 2 after having a primary infection. But we believe right now that it's low risk that you would be reinfected after 3 months. Another study showed that after about 5 months, you're about 83% less likely to get an infection. And so I think we just need more time to understand the immunity and how well our immunity is going to endure. And then severe COVID, this is so interesting that how this virus works. I think we're only scratching the surface right now, but even people who just have mild outpatient disease are still having prolonged issues. Someone I work with closely, they got COVID, they're really great shape and are still struggling from breathing issues. So they can't really run for long distances. This is someone who has ran marathons. They're in really good shape. I wish I was in the same shape as them. But it's important to know that just because you think you're young and healthy doesn't mean that COVID can't affect you and hurt you down the road. The other one is asymptomatic transmission. So asymptomatic transmission is very, I really don't know what to say about this topic. These are numbers that were reported in a recent publication that's 59% of transmission comes from asymptomatic transmission. And that, maybe fomite transmission is estimated to be about 10%. I feel like a lot of these are just random guesses. It's our best guesses based on modeling. But I think what the key takeaway here is that people can transmit while they're asymptomatic or pre-symptomatic. And that's something that we weren't seeing with SARS coronavirus 1 very often, and is something that's very challenging for us to wrap our arms around and control this pandemic. All right, so I'm going to shift gears and talk about vaccines. Hopefully I don't need to spend too much time on this topic. Hopefully everyone here understands that vaccines do have a positive impact. If you look at in the US population, it increased from 76 million to 279 million between these years. Life expectancy rose. The top 3 causes of death are no longer infectious disease. And it's not correct to say that all of this is due to vaccination, but vaccination did play a critical role in this. And vaccines have been around for a while. We have about 27 we have vaccines for 27 diseases. We have more than 27 vaccines overall, but vaccines do have a positive impact. You can see just how the measles vaccine and polio vaccine have driven down these infections and disease. All right, and so types of vaccines. This is where I think people kind of get lost in what is each vaccine doing. So there's an inactivated version. So if you've had the hepatitis A or an influenza vaccine or polio vaccine, this is a virus that has been inactivated by some form or fashion, maybe formaldehyde. There's attenuated vaccine. So if you had like the measles, mumps, rubella, or another type of polio vaccine, you've had an attenuated vaccine. Viral subunits are basically just pieces of the virus that elicits an immune response. So these would be antigens that we hope elicit a strong immune response. And these are generated in several different ways. And then the last one that is relatively, I don't want to say new because it's not new. We've been studying nucleic acid-based vaccinations for a while. It's just the mRNA vaccines that we're using are new to the market. And we'll talk a little bit more about these on this next slide. So for SARS coronavirus 2, Pfizer Moderna vaccines, they use a nucleic acid-based approach. So this is mRNA. Remember, mRNA is messenger RNA. So they have a synthetic message, and this is the synthetic message. It is very delicate, and if you look at mRNA wrong in the lab, it degrades like that. It's very quick. It's kind of been the bane of my existence in some settings, but some very smart individuals encase this in a lipid nanoparticle to protect this messenger RNA. And so when they inject it, it gets injected into your arm. And then hopefully what happens is you break some muscle cells, the muscle cells respond, your innate immune system responds, and then hopefully that gets funneled to your lymph node, most likely underneath your armpit, and that's where your B and T cells will start to have a reaction to it and generate a more robust immune response. And as they generate that immune response, your lymph nodes will begin to swell. That's why doctors always check your lymph nodes to see if you may or may not have an infection. But what's happening in the cell is the spike protein, or sorry, not spike protein, the messenger RNA gets put into the cell, and the host cell begins to spit out the spike proteins, and then those will hopefully be presented on your immune cells, those antigen presenting cells, which then can stimulate your immune response. And so in this, there's no, I don't want to say it's impossible, but this is not really being put into your nucleus where it can integrate into your DNA. There's something called a poly A tail that is associated with this that prevents that from being pushed into your nucleus. For any of those any nerds out there who understand that, but there's very low risk of that ever happening, and these cells that are being infected are dying within a few days too. So it's not like they're getting into cells of concern. All right, and so for COVID-19 vaccines that are currently out there or about to be out there, the ones in green are the ones that I'm going to talk about specifically. There's 36, or there's over about 100, or I think it's 90 last time I counted, 90 vaccines that are in some sort of trial. There's 7 that are authorized, and this is globally, not in the United States. So there's a lot of people out there trying to fight this, and obviously there's some clear winners because they made it to market first, and those are the ones that are in green, or I shouldn't say they're all in market, but the Pfizer, Moderna are in market in the United States, and Johnson & Johnson hopefully will be by the end of this week. All right, so Pfizer and BioNTech. So this is an mRNA vaccine. So we talked about what mRNA vaccines are already, but this mRNA vaccine will express the SARS coronavirus 2 spike. So when it's doing that, it's only expressing this spike protein. So nothing else is being expressed. So you're not generating virus, you're not generating something that's going to go on and infect you further. It's just that antigen. It's a 2-dose schedule. Day 1, day 21. The vaccine storage is a little unfavorable. When I say a little, a lot, and it's negative 70 degrees Celsius. So that's kind of a struggle for them logistically, but they're still doing it. I also say that the UK, in some cases, has switched to a 12-week dosing schedule, and we'll talk about that a little further in this presentation. But their vaccine interim results demonstrated a 90 and 95% efficacy. So really, really good efficacy. And the point here is that this is efficacy data, not effectiveness data. And these are also interim analyses. So we might find out in 2 years from now that, hey, when we looked at the totality of the data, it's actually 88% or 89%. So right now, what we know based on current infections within their phase 3 trial, it looks like it's 95% efficacious. And also, I like to point this out too, that it's not like these vaccines are done, that you got the results. There's a lot more that goes into these vaccines. And this is how Pfizer and BioNTech are doing theirs. This is you can see out here at 24 months, they're following up with these individuals 2 years out. So the study has only just begun. And the same is true for the other vaccines, as you'll see. Moderna, so this is a very similar vaccine. It's a mRNA vaccine expressing the SARS coronavirus 2 spike protein. It's the pre-fusion spike protein, and it's a 2-dose schedule, slightly different, but the vaccine storage conditions are more favorable because it's only minus 20 degrees, and you can actually store it at refrigeration temperatures for 30 days. And then so for their phase 3 interim analysis, same thing as I said for Pfizer and BioNTech. It is an interim analysis, and they show really good efficacy results. But again, their trial is 759 days long. It's you're kind of in this period right here, and so they have a lot more that they want to learn. And when I say that they have a lot more that they want to learn, obviously they're looking at safety, but they're looking at immunogenicity and understanding how our immune systems are responding and whether or not maybe we need a booster, maybe we don't need a booster. There's a lot of stuff that's being evaluated here. AstraZeneca, University of Oxford, this is being used in other countries. It is not yet available in the United States. I think the FDA is a little peeved with them. And I guess rightfully so. Their trial didn't go according to schedule. So they have a vaccine that delivers a recombinant replication defective adenovirus. It's a chimp adenovirus, but it also expresses the SARS coronavirus 2 spike protein. They do a 2-dose injection schedule, day 1, day 29. The best part about this is it has a very favorable storage condition, 2 to 8 degrees Celsius. They don't have to store it at super cold temperatures. But their initial interim results showed that there was a 90% and 62% efficacy. And so this was on the 4th of November. And what they found was there was an accident in how someone was calculating the amount of virus in the vaccine dose. I think one was using a spectrophotometer and, or sorry, not a spectrophotometer, a mass spec to analyze the concentration, and another lab that was contracted was using a separate method for calculating the concentration. And because of that, there were 2 vaccine dose concentrations. And what they found, which is different than what they were attempting to do, is that the first vaccine had a smaller dose and it was followed up by a full dose. And that actually elicited a 90% efficacy, whereas the standard full dose, or standard dose elicited a 62% efficacy. I mean, there's a lot of reasons why this may have happened. Maybe the antibodies got rid of the vector on the second one. This could be a numbers/statistical issue because the group was a lot smaller that received the small dose followed by the standard dose. And there's also some issues with how the vaccine was delivered and the dosing schedule. And that's what this is the fourth bullet. Fourth bullet is getting at. They actually had different dosing schedules that were, I think, on accident as well. But they found those who were getting dosed, and again, this is from data on the 7th of December, those who were getting dosed with a standard dose and a standard dose at greater than 12 weeks, so not following this day 1 to day 29 protocol, found at 82.4% efficacy compared to those who got a less than 6-week dose of standard dose. And that was 54.9%. So they haven't filed for EUA to my knowledge, but I think the FDA is just a little confused and scratching their heads about what really happened with your trial. Things didn't really go as planned. And so I think they're really wanting to understand that before they grant anything. Johnson & Johnson, fingers crossed that this gets approved. I think it will. The FDA came out, I think yesterday, and said that it looks good. They'll review it, or their independent panel will review it on the 26th. They showed that you get 66% efficacy after 28 days. That's moderate to severe. 85% for severe disease after 28 days. And then they showed 100% efficacy against severe disease after 49 days. So really good results, really favorable storage conditions, similar to AstraZeneca. And it's a single dose vaccine, which is really great. I know some people don't want to go through 2 doses. I personally got the Moderna vaccine, the first shot, and it's kind of a pain to schedule the second one. All right, Novavax, this one is a smaller trial. They only had 15,000 folks involved with it, but their initial results are good. And that overall 89.3% efficacy. They did show that those who were tested in UK that had the B1117 variants, it was 85.6% efficacious against them. No timeline on when they would file for an EUA, but this is another new type of vaccine. It's a viral subunit platform. So when you look at it, it's almost like a little flower with the spike proteins. And I'm not sure how much the United States has secured for this vaccine, but there's a lot that I think will be coming on the market in the next 3 months. And hopefully we can get everyone vaccinated and put this behind us, and get on with our lives and get back to real food safety problems. And so who gets what? I think this is a really interesting graph I won't spend much time on, but you can see who really jumped the gun and tried to secure doses quickly. So you can see, a lot of your first world countries secured a lot of doses for their population, but then there's also some equity issues. I think there's a lot of countries who you'd expect to get vaccine for their population didn't really jump on the wagon as much, and some of these numbers might, if you look at China, for instance, China may not have secured a lot, but they're also creating their own vaccine. So there's, you kind of have to look at every country in its own lens. All right, and so are vaccines safe? This is a really important question, and I think it's an important question that everyone asks. No medical product is completely safe. There are side effects associated with every medical device and medical treatment that needs to be talked about. But I think the track record for vaccines overall is that they're low risk of severe side effects. So in the United States, we have the Vaccine Adverse Event Reporting System, and then we also have the Vaccine Injury Compensation Program to handle this. So if you look at from 2006, 2016, there's over 3.1 billion doses of vaccine distributed. And so there's about 30,000 reports per year to the Vaccine Adverse Event Reporting System. And most of these are actually what may be considered as reactogenicity, so mild side effects such as fever. And so if you wrap this all up, this is about a 1 in 10,000 chance of a reaction that you may report to virus. And then, it's important to talk about adverse events because they do happen. It's no secret, but people do get compensated for any adverse events that may cause injury. So from, I'll just go with the bottom one, 1998 to 2018, there were 19,000 claims. And among those, about 1,200 deaths. And so about 11,000 of these were actually dismissed, and then 5,800 were awarded some compensation. So vaccines, again, they're not completely safe, but they are very low risk, and I think taking a vaccine definitely outweighs some of the repercussions of trying to develop your own immunity through natural infection. And a really classical example is for smallpox vaccine. It carried a 1 in 1 million chance of death from receiving the vaccine. But if you wanted to roll the dice, you had a 1 in 30% chance of dying from the disease. All right, and so vaccine timeline and safety. We're kind of here. This SARS coronavirus 2 pandemic, really forced us to think outside the box for getting vaccines in people's arms. So we're sort of in this area right now. And people ask the question, is it safe to take this vaccine? And so what I've told folks is that it's an EUA, so emergency use authorization, which I guess people kind of are skeptical about. And then also people using the word warp speed and like race to the finish line. It makes it seem like there's people cutting corners. But I would say that one thing that is concerning is the length of time for measured efficacy. And I would say that none of these vaccines are licensed yet, because no one's going to get a licensed vaccine with only 3 to 4 months of efficacy data. I think mumps was the quickest vaccine to licensure, and that took 4 years. But I don't think anyone would raise their hand and say, yeah, let's wait 4 years or 5 years or 6 years or 7 years. So we fully understand this vaccine before we start immunizing people, given that we are at almost 500,000 deaths in the United States. HPV took 7 years. So there's reasons for waiting, but under the conditions, I think it is appropriate for an EUA. And then the other piece is size of the trial. I think size of the trial isn't so much a concern. I've heard Paul Offit talk. He's kind of a god among vaccinologists, that Moderna is 30,000, Pfizer had 44,000 participants. These are normally sized trials. And then when it comes to safety, most safety significant safety issues occur within the first 2 months of dosing. So we're well past that, and there haven't been any severe, serious side effects attributed to the vaccine. If you think of some of the big incidences like the Cutter incident or the intussusception issue with RotaShield, or reduced platelets associated with the measles vaccine, or Guillain-Barré with flu vaccine. Well, no, I won't say Guillain-Barré, you can strike that. But the other ones, those all really happened within the first 2 months. Flu vaccine can be associated with Guillain-Barré syndrome, and it may pop up after 6 weeks, but there's it's like a 1 in 1 million chance. And we also recently found out that a traditional influenza vaccine can also cause Guillain-Barré, so it's not really vaccine related. But so yeah, safety is a key piece throughout this timeline, and the reason we're getting this vaccine so quickly is because the red tape's been cut, and there's literally been billions of dollars poured into this, whereas most vaccines kind of get stuck in this phase just looking for funding, and that's partly why it takes so long. All right, and so we'll stop there and move on to variants. This is kind of what people are concerned about now. Hopefully I can stop after this and take some questions in the last 10 minutes. But so SARS coronavirus 2 variants of concern. I'm personally not so concerned by them. I think they're pieces of the information or pieces of the puzzle that we as scientists need to really understand, because it will inform our pharmaceutical interventions. But yeah, this isn't something that we as a society should just stop and freak out over. This is from Vox.com, published in early January, and it was talking about the variants, and maybe we need to stop going to the grocery store. I think this type of stuff just really infuriates me, because we're trying to create pandemonium from the media, and it's really not necessary. So right now, there's no evidence that these variants cause more or less disease. And right now, there's no change to the pharmaceutical intervention recommendations. And I say yet because there is some early indication that monoclonal antibody therapies may be impacted by these mutations. And so this picture here is an example of what I'm talking about. So when I defined antigen, I said it's an immune stimulating material. But within this material, there's specific points that are immune stimulating. So this is just a simple diagram that shows what we call epitopes. These are very specific pieces that elicit the immune response. And so, here's a triangle one, here's a circle, here's a box. So what I say when I say monoclonal antibodies might be impacted, well, these two might still work, but because of one amino acid change, this antibody might not be as efficacious as previously thought. But there's also other antibodies that, or monoclonal antibodies that would work against other parts. And the benefit here too is there's, I think, 20 T and B cell epitopes on the antigen, or sorry, the spike protein that is the antigen we're vaccinating against. And it's really easy for companies to pivot their monoclonal antibody therapies. And same with the vaccines too. Moderna is already updating their vaccines and looking at that to include the, I think it's the B135 variant that was originally identified in South Africa. And they said they can update their vaccines in 2 months. And that's the benefit of doing mRNA vaccines. They're very nimble. All right, and so I went to the CDC website and also looked at some other sources to help put this together. But there's 3 variants of concern that we're really looking at. And there's one that was first detected in the UK, Brazil, and now Japan and South Africa. These are the key mutations, but these are really the concerns associated with those mutations. D614G, that's suggested to increase transmission. I don't really buy into this one very much. It was identified very early on, and people were thinking, oh, it's a new strain, but I don't think that it's as much of a concern. The N501Y mutation, this is suggested to improve binding to ACE2. And then these 2 last ones may affect the monoclonal antibody therapy. So there are key changes. I think what we're observing here, based on the amount of sequencing that we're doing, it's really awesome that we're doing this. Hasn't really been done before at this scale. But I think what we're seeing here is the beginning stages of what can be considered antigenic drift among SARS coronavirus 2. But again, really important for scientists to look at and maintain and keep a watchful eye on. But in no in no world do you need to stop going to the grocery store because of the variants. And there's no real change to non-pharmaceutical intervention recommendations due to the variants. So still follow CDC guidelines, social distance, wear your masks. And I think that's it. So I guess I'll pause here and see if there's any questions. I know I went through a lot at once, but there's a lot of information to cover, and Jason's going to record this so you guys can listen to it later. But yeah, I'll take any questions if there are any.
Yeah, there's some good questions in the chat here. The first one is, what data have you seen for the use of a face shield in addition or instead of masking?
Oh, I have seen data. That's a good question. I need to look that up again. There is one paper that looked at it. It might have come from an MMWR, and I kind of chuckled because I think it was 1%. Don't quote me on it, but it was extremely low to the point of why wear it. I'm not sure why anyone ever thought wearing just a face mask, or sorry, not a face shield, why that would be of any use, but using it in conjunction with the face covering is good. I think face coverings in general, I've seen about 50% from the data that they're helping about 50%. That's what I found for the face coverings. But there's a lot more research we need to do on face coverings in general to understand their true impact. We know that they have some benefit, and it's reasonable to ask someone to wear a mask, but yeah, we need to learn more about the true benefits of masks.
Okay. The next one is, what are your thoughts on the perception that as soon as a person receives the vaccine, they no longer have to observe mask wearing, social distancing, and other mitigation efforts?
That's a good question too. We're still learning about what happens to you after you've been vaccinated or after you've been immunized. So everyone's different. We can't make a blanket statement and find one individual, one anecdote, and make a determination off of that. But I think if you've been vaccinated, the likelihood of you being reinfected is very low, but it isn't zero. And so trying to get someone to say, oh, I'm not going to wear a mask for 3 months. It's really hard from a public health standpoint to tell people, oh, don't wear a mask because it's probably low risk. You don't really know that. And my personal belief is that our vaccines are going to do well at preventing disease. They may have some protection against preventing infection, but not as well as some other vaccines like the measles vaccine. So I think if you've received the vaccine, it's still important to wear the mask because you don't know who hasn't received that vaccine yet or has had the chance to get the vaccine. And you don't want to, you're protected, but they're not protected. So if you get the vaccine, there's still a chance that you might be infected and may still transmit the virus. And I think that is out of an abundance for precautions because we don't have all the data to state otherwise. So it's really out of abundance of caution.
That's good advice. The next question is, am I understanding that if I receive the vaccine, I become immune to the symptoms or to the disease? Can I carry the virus and transmit once I receive the vaccine?
Yes, so there has been evidence of people being infected after they've received their vaccine. And there's no surprise here that this happens. Our primary endpoints for all of the vaccines that are being evaluated right now is, does the vaccine prevent disease and different levels of the disease? So severe disease, moderate to severe disease, mild disease. And what we're really trying to prevent is severe disease. So, if you get mild disease like the sniffles, maybe a headache, maybe a mild fever, but it prevents you from staying out of the hospital, that's still a win. So if you do get the vaccine, what we know now is that you are being protected to some level against developing a type of coronavirus disease. So whether it's mild, moderate, or severe, and we're still waiting to hear whether or not this vaccine prevents infection. I think there's some indications that it does help with infection, but we're not sure what that is. Is it 10%? Is it 100%? We don't know. My speculation is that it does have some benefit to preventing infection, but I think it's going to be pretty low, whatever it comes out to be.
Okay. The next one is, after being vaccinated, is protection permanent, or will people need to be revaccinated?
That's the $1 million question. A $1 million question for Pfizer, Moderna, and all these other vaccine producers. Right now, they might not have a need or a financial benefit to license these vaccines. They may just say, hey, let's, the data may tell us that people are getting the vaccine and they're expressing durable immunity that lasts for a very long time. And so we don't need to do that. I don't think that that's going to pan out the way that I just described. I think that, we will need a booster at some point. And that's based on some recent evidence I've listened to and read about, which is that SARS coronavirus 2 does evade our immunity through antigenic drift. And so if you look at regular human coronaviruses that circulate year in, year out, they do evade our immunity. And so this is maybe like 4 to 6 years, but that does happen. So we can't expect that this virus isn't going to mutate and evade our immunity. So I don't have a hard answer on that, but my guess would be that in the future there is likely going to be a need to get a booster. But the CDC is still looking at this. There's no definite answer on it, but yeah.
That's great.
Maybe this time next year we'll have a better indication on when you may or may not need a booster.
Yeah. The next question, is it safe for people to travel after they have been vaccinated?
Yeah. So if you think about what's okay for you, maybe there's some countries that don't even want you to come over, but I think within the United States, if you've been vaccinated, I'd say still wear your mask. I think it's okay for you to travel because you are going to be protected. But again, you don't know if you may be reinfected and you may be asymptomatically transmitting that to other people. So just keep that in mind and still follow the same precautions if you do travel. We can't lock down everyone for the rest of their lives because of this virus, but yeah, there's good evidence to say that it's okay to go out and start traveling somewhat, but still, keeping a watchful eye on what you're doing and still trying to social distance because of our unknowns about reinfection and transmission.
We are at time. Do you have time for one more question?
Yeah, sure.
Okay, the last one here is piggybacking on the face shield question. Are the eyes a route of transmission being a mucosa? Could droplets fall in the eyes and act as a route of entry?
Yes, absolutely. Yeah, that's something that people kind of overlook. And I would say I overlook too. Whenever I go out, I'm not wearing eye goggles. But yeah, this is something to consider. I think that the transmission through your eyes is probably a more challenging way for the virus to establish infection compared to going straight into your nose or your mouth. But it is certainly a way to be concerned about. And if you look at just like our company policy, back in July, we were recommending that everyone wear a mask, but we also asked our employees who were going out to customers and working with customers and are at more severe or more risk than someone like me who works in an office. We started asking them to wear glasses and goggles to help protect against that type of transmission as well.
That's great. Thank you so much. I'll hand it back to Jason.
All right. Well, thank you, David, for a terrific talk. Something, again, we'd like to thank our sponsors and certainly all of you for participating. We'd like to kind of show you that we have our next seminar is planned for March 17th. We have Kim Livesey and Shelley O'Toole, and they're going to be talking about some of the other efforts in terms of food distribution that have occurred due to COVID. Again, we have this. This is going to be recorded. It'll all be available on the FAFP website. So very easy, fafp.net. You can just go to the events and education, and that's where we have actually this year's webinars as well as last year's webinars. So please go check those out and share widely. So again, thank you, David, for a great presentation, and hopefully we'll see you all next month.
Yeah, thanks everyone. It was a good talk. Good questions.